It's an honor and a privilege to actually stand over here a year after the CVGPS was announced to share with you how the Framingham Heart Study and Boston University will operationalize the mission and the vision and the scientific foundation that Dr. Loscalzo elegantly described. I have no conflicts of interest. I do want to disclose that Boston University receives a modest amount of funding for participating in this and for facilitating the grantees. This is a slide which probably some of you saw a year ago at the scientific sessions, and it describes something that Dr. Loscalzo elucidated, the combination of different stakeholders who are participating in CVGPS. My own presentation is largely going to focus on the Framingham Heart Study and Boston University aspects of CVGPS. And I hope to walk you through over the next 10 to 12 minutes 4 important elements. One is just the design of the Framingham Heart Study, so that some of the fellows and the young investigators in the audience can understand the wealth and the design elements of the Framingham Heart Study that lends it unique to CVGPS. Then talk a little bit about the plethora of resources, scientific resources that the Framingham Heart Study has, which will be available through the CVGPS and through other initiatives as well. Then I want to spend a little bit of time walking the people in the audience through the ancillary study policies at the Framingham Heart Study, because we have used CVGPS to adapt the existing ancillary study policies, so there's no internal conflict between the way Framingham conducts business on ancillary studies as part of CVGPS or outside of CVGPS. And the last part, if you're curious about what exactly the Framingham study and Boston University does for the Framingham review, I'm going to share with you the elements of the review process within the Framingham Heart Study. So this slide displays for you the 6 different cohorts that constitute the Framingham Heart Study. 3 of these cohorts are multi-generational. The original cohort, beginning in 1948, about 5,000 participants have completed the 32nd biennial examination for up to 64 years of follow-up. The children of the original cohort and the spouses of those children constitute the offspring cohort. Another 5,000 people have completed 9 quadrennial examinations or 36 years of follow-up. And the 3rd generation cohort, about sample size of about 4,000, have completed 2 serial examinations. At the time of the 3rd generation cohort, we went back and actually recruited some of the offspring spouses whom we had missed to recruit over there, just a modest amount of sample size of 100. So these 4 studies constitute the multi-generational Framingham Heart Study, and these are predominantly white individuals of European ancestry. There are 2 other minority studies that are part of the Framingham Heart Study, the Omni cohort and the Omni cohort second. These are not related to each other. In the next slide, I'll try to capture for you the important elements of these 6 cohorts. I'll begin with the minority cohorts, which was what I was talking about in the last slide. And you can see that this is a modest size minority cohort, and about 40% of these cohorts are Hispanic Americans, and about a quarter are Asian Americans or African Americans. So this is a nice minority cohort that serves as a reference within Framingham, and also was recruited so that the demographics of the Framingham sample reflects more of what the town of Framingham has evolved into over the years. The other 4 cohorts, and as you can see, the total sample size across this adds up to about close to about 15,000 individuals, examined from anywhere from 10 to 65 years, with a wide age range at the time of enrollment. One of the lesser known facts about the Framingham Heart Study, and this is to share with you because CVGPS is a genome phenome study, is that the Framingham Heart Study is a family-based study. Nested deeply within the 3 generational cohorts are large pedigrees, something that often we don't talk about when we analyze Framingham data, but which we pay meticulous attention to. And you can see somewhere between 6,000 to 8,000 first to 3rd degree relatives are nested within the Framingham Heart Study cohorts. And this is like a geneticist's delight to really look at the segregation of, whether it's segregation of rare variants or familial transmission patterns. The Framingham study lends itself uniquely to this purpose. Let me segue now onto the kind of resources available within the Framingham Heart Study, which is open to all applicants of the CVGPS. And 4 important points just to emphasize about the Framingham Heart Study. The Framingham Heart Study is like the mini human phenome project. And in my next slide, I'll illustrate why I say this. And we measure multiple traits in multiple cohorts at multiple time points. And this covers the entire spectrum of lifestyle measures to novel risk factors. So that if you really want to look at the time average measures of risk, if you want to look at the slope of risk factors, Framingham has it right starting from 1948. If you want to look at temporal trends in these trajectories, we have the data. And then it's a family study. And I just covered in the previous slide the large number of pedigrees that are nested within the three-generational family structure. And we have extensive genetic and genomic resources, which I shall summarize in the subsequent slides. So we call it the Framingham Heart Study. But I can tell you it's more like the Framingham study, because there is the Framingham Stroke Study, the Framingham Dementia Study, the Framingham Bone Study, the Framingham Arthritis Study, the Framingham Eye Study, the Framingham Hearing Study. I could talk for another 30 minutes and tell you all the various elements of the body that the Framingham study covers. So it's a true mini human phenome that has accumulated over the years. At the bottom of this slide are the omic resources, and I shall highlight these subsequently, made available through an NHLBI-supported project called SABRE, championed by my colleague Dan Levy, who's over here somewhere in the audience. And I also want to highlight the importance of the CMS data. If you're interested in medical diagnosis of various kinds, Framingham participants do have CMS data to which you can link a lot of medical diagnosis, making it really a scientist's delight. When you talk about phenotypic characterization in Framingham, what this slide shows are the three kinds of outcomes that we really have a rich data set on. We have the hard clinical outcomes that we all are interested in, but we have an extensive repertoire of subclinical disease measures. Almost any part of the heart or the vascular system that could be imaged, be it the peripheral arterial system, the medium-sized muscular arteries, or the large vascular elastic arteries, they have been imaged. It could be coronary calcium, it could be brain MRI. And we have a plethora of traditional as well as standard risk factors and novel risk factors that we have acquired. What this shows over here is if you go to framinghamheartstudy.org and click on the tab for researchers and download the Excel spreadsheet, you'll find a large Excel spreadsheet that sort of tabulates very, very carefully the three kinds of data that we have. The non-invasive test that includes non-invasive imaging tests as well as lab tests, as well as those acquired by the physician examiner and questionnaires. And you'll find the data set number and the type of cohort and the time interval. And you'll find a coding manual and annotated form. So please make use of this. We are always there to help you. A little bit about the extant and genotypic resources. The Framingham study has been at the forefront of population-based genetic research right from its inception. Every time a new technology came, Framingham was adequately and prominently represented, be it the microsatellite markers that were the basis of linkage studies, or the candidate gene care project, or the large-scale whole genome-wide approach, the SHARE project. More recently, as part of the CHARGE consortium, we have developed data sets on the whole exome sequencing and the low-pass whole genome sequencing. Through our intramural scientists, Chris O'Donnell and Dan Levy, we have exome chip on 2 of our generations. And more recently, Framingham Heart Study is funded to acquire whole genome sequence, 30x or more, on up to about 4,500 heart study participants, which will be available in about 12 months in an NHLBI data commons as part of a premier NHLBI whole genome initiative. We are very proud to be part of this initiative and hope that this opens up the door for a lot more investigation of genome-phenome relations. A little bit more about the genetic resources. It looks like a complicated slide, but this just captures the data that you had in the previous slide. And I want to focus on the parts in red because these are the most updated data sets. As I mentioned, we have got whole exome sequencing available through a couple of projects. And we have got whole genome sequencing available low pass through the CHARGE sequence project. And we have targeted sequencing as well available on a set of participants. So what this slide again shows is the kinds of omic resources. Dr. Rascalzo spread out very nicely in a slide the elements of the systems biology, and Framingham is well represented in several of these. We have whole exome sequencing on close to about 3,500 participants. This number is a little bit old. We have whole genome sequencing on up to about 1,200, low pass whole genome sequencing. Like I said, we'll have whole genome sequencing through an NHLBI initiative on about 4,500 individuals. Gene expression facilitated by the SABRE project led by my colleague Dan Levy on about 5,600. The methylome, again, it's a project facilitated by Dan Levy on about 2,700 people. The metabolome, there are multiple sources, including a collaboration with Robert Gutson at the Broad Institute, where we have the metabolome on about 2,500 people, circulating microRNAs, and a plethora of protein biomarkers on nearly 7,500 people, all available for your analysis. This is a slide borrowed from my colleague Andrew Johnson, just to tell you and show you with different colors that every time a new technology came in, Framingham was right there at the forefront. Shown here in red are the heritability of family history studies, the linkage era, moving on to the candidate gene study, and more recently the era of whole genome sequencing and multiple consortia. So all these data sets are there available. A lot of the data are available in dbGaP, and they are available for your research. A little bit about the ancillary studies policies at Framingham. And the key principles here are that we'd like to protect our participant confidentiality. We want to respect their informed consent. We want to limit participant burden. And we want to ensure the best use of precious, non-renewable resources. With these four key principles in mind, we have a complicated system which we have sort of simplified over the years. And shown here in red are the two specific committees, the laboratory committee and the DNA committee, with which CVGPS grantees might have to interact. And I'm going to focus a little bit on this. So when you apply to the Framingham Heart Study through the website and you fill in an application form, this I'm talking about all applications, not necessarily CVGPS, it's reviewed by our committees. And then after approval, you get a data and material distribution agreement which is executed. And we track the materials that we distribute. And then if required, and if there's a Framingham element, it's integrated into the Framingham team. The project is conducted. And then finally, the data are returned back to the parent study so that they can be shared with the worldwide group of researchers. So what does Framingham do as part of CVGPS? The AHA sends us the application. We complete the initial review through a Framingham Heart Studies core screening panel. It's important to emphasize that since Framingham investigators are eligible for application, this core screening panel includes individuals who do not apply for CVGPS, including myself. We see whether it requires review by the DNA or the lab committee. If the data are available in BioLink or dbGaP, it does not require this review. If not, we go through these committees. We have a final review, and we send the results to the AHA, and the AHA selects the awardees. One important thing is, what do we look for? We look for feasibility, overlap, and participant burden. The Framingham Heart Study does not score the grants. That's the job the AHA does. We just talk about these elements. And on the right side of this last slide, we have an overview of the various committees that we integrate this to really make this possible. So I hope several of you in this room are applying for the grant challenge. I hope to see several of you in this podium a year from now for the pathway grants. Thank you.